One type of drug is also used to prevent the HIV infection. It is a lost option. It, but it is very complicated to understand this drug. Uh, but anything is not impossible for human beings. So let's see here. This is the DNA formed by the reverse transcriptase present in capsid of HIV virus inside the host cell. When first see here, the reverse transcriptase first form DNA and then cut RNA molecule, then it form the double helix of DNA molecule, and after it by the enzyme, by the help of enzyme integrate the DNA molecule of HIV enter into the genome of a host cell, and after entering it, it forms some proteins. See here, these proteins are very important. So I zoom this structure here. See here, this is the structure of hiv hiv dna. There are three segments in hiv dna. Three segments are three gene present in hiv dna. First one is gag gene, gag gene. Second one is pool gene, and third one is env gene. These three genes are present on hiv virus dna, and gag gene synthesize the p24, and pool gene synthesize reverse transcriptase integrate enzyme, and third one is polypeptidase, and env gene synthesize glycoprotein 120 and glycoprotein 41, and p17 is also synthesis by gene. First say this is the gene, three gene present in hiv viral dna. These are synthesize these types of protein. Gag gene synthesize p24. P24 is a protein which is present on nucleoside, which is present on capsid. See here, this is p24 synthesized by gag gene, and p17 it is also present in, here p17 matrix. It is also synthesized by gag gene and pool gene. Pool gene synthesize three enzymes present inside the capsid protein. These are retrovirus, and second one is integrate retro retrovirus, no, reverse transcriptase. Second was in one is integrate enzyme, and third one is polypeptidase enzyme. These are synthesized by pool gene, and env gene synthesize two proteins, gp120 and gp41. Gp120 is the outermost protein which first attached to the CD5 molecule, and P41 is a integrated protein which help to combine the cell membrane of host cell and HIV cell. These proteins are synthesized by these three genes. But we have the drug uh, known as a protease inhibitor. What is the function of this drug? This is a group of drug. See here, the function. What is the function of polypeptidase? Polypeptidase, when these are formed, first they contains only one chain. These are only one chain. These are only one chain. Polypeptidase cut this into two segment, this into three segments, and here polypeptidase uh, also work, but it is cellular polypeptidase, not viral polypeptidase, into two segments, not three, two segments. Polypeptidase cut into P24 and P17, cut into reverse transcriptase, integrate, and uh, polypeptidase. Before cutting, these protein are non-functional. When polypeptidase not cut, if polypeptidase not cut this protein, this protein molecule will synthesize by these genes, but these are non-functional. So when polypeptidase cut these enzymes into two or three segments, then these enzymes are functional. So what is the drugs function of protease inhibitor? Protease inhibitor inhibit these protease polypeptidase enzyme. When polypeptidase enzyme inhibit, then this non-functional protein will not cut into functional protein. So non-functional to functional protein not occur. Therefore, after synthesize all these things, uh, the genetic material or gene or protein of HIV will not go further for transcription or go to the cell membrane for rupture of the cell membrane. So this is the last drug. What are the drugs? The drugs are indinavir. Second one is duranavir, and fourth one is sakinavir. These Vir drugs are used in protease inhibitor or polypeptidase inhibition. Indinavir, retonavir, and duranavir, and sakinavir. So these are drugs. So drugs are very important in our syllabus, uh, and I want to combine all drugs in one place. So see here, drugs use in HIV suppression. Number one. Number one drug which is used in HIV suppression. This is Meraviroc. Number two, Meraviroc bind with CCR5, so P120 will not bind with CCR5 molecule, and infection will not occur. And Infuortide. Second one is Infuortide. Infuortide inhibit P41 inhibit it suppress CCR5 inhibition. And the third group of drug is nucleotide reverse transcriptase inhibitor. There are two types of drug. First, form nucleotide without hydroxy, and the second one is inhibit the reverse transcriptase. So which form nucleotide? These are the adenosine. Second one is lamivudine, and third one is zodivudine, and which inhibit the reverse transcriptase. These are uh, nevirapine. This is nevirapine. Only one drug is here, and these drugs inhibit the reverse transcriptase. Fourth type of drug, uh, it is raltegravir. Raltegravir. The function of raltegravir, it inhibit uh, inhibit integrate enzyme, and fifth one group of drug, it is protease inhibitor. Inhibit the polypeptidase. So these drugs are used in suppression of HIV infection. We complete our six topics. The last one topic, cause of vaccination failure. Why vaccination is not occur in HIV? Uh, See here, this is a P120 molecule. P120 is synthesized by gene uh, known as pool gene. Pool gene synthesized P120, and it is very mutant in very less time. So when P20 when a pool gene will mutant, there are some modification occur in the P120. So if we form antibiotic which is star antibiotic, here is star antigen, and we form a star antibiotic. After some time, it go further modify, mutation occur, and form moon antibody, moon like antigen. So when moon antigen, this antibody, star antibody, will not act on this antigen, so this protein will not rupture or not dysfunctional. This is the cause of vaccination failure, because P120 is highly variable. Within some times or some weeks, it will be changed. Scientists form many types of antibiotic. About 20 types of anti-P120 was formed by our scientist, but more than mutation occur. Therefore, this is not functional. And there is one question: when P120 become mutant, then how it will be attach on CD4 molecule? The answer is that the mutation is it is very clever molecule. So the mutation is not occur on the binding site. It is mutation occur on another site where the antibiotics or vaccine uh, bind. So on the binding site there is no any mutation occur, so there is no any problem with the attachment with a CD4 molecule or CCR molecule. So this is the reason of failure of vaccination uh, against HIV infection. So we have complete our today's topic. What is AIDS uh, and structure of HIV, how entering into host cell. The main thing is that it enters into by the help of P120, GP120, and replication in host cell, how it occur by reverse transcriptase, and uh, the next is a what is a test for AIDS or HIV infection. These are ELISA, PCR, and number 5, 3, 6 drugs. These are many types of drugs we discussed briefly, and the last one, cause of vaccination failure. Because of it is high, GP120 is highly variable, so there is no any permanent solution of vaccination about GP120, and vaccination will fail after some times. So thank you.